thank you very, very much for inviting me here. It's been a long time since I've been to this campus. Dan and I were trying to figure out when I was last here, and it's been at least 10, 12 years since I've been here. So it's nice to be back. And this really is a wonderful honor to be invited to be the distinguished visiting ecologist. I've never had such a title before, so thank you very much. And thank you all for your hospitality today and also tomorrow. And I met some of the students today, and I really look forward to my meetings with the students tomorrow and learning what you guys are doing as well as what you think of these papers. So I'm going to give two talks based on two things we've been working on, but there's a common thread between the two talks, and that is delays in asynchronies in ecosystems. And today I'm going to talk about some work that we've been doing about delays in the nitrogen cycle and population dynamics of wild rice and complex behaviors of the populations of wild rice. Tomorrow I'm going to be talking about delays and asynchronies in a model of chronic wasting disease. But the difference is, in this talk, the idea of delays was built in right at the beginning in our hypotheses, whereas in the talk tomorrow, when we started that modeling work, we didn't realize that we were dealing with delays until the very end, and we're trying to figure out why the model did something.
Delayed realization.
It was delayed realization on our part. So lately I've been thinking a lot about delays and asynchronies in ecosystems as a whole. Okay. Well, I'm trying to figure out how this, how do I get this to move? Oh, maybe it's that one.
I don't know, it's an Apple, and who knows how they work.
It's the arrow down, or? Well, it's not moving it.
This takes director level intelligence. Bit off more than you could chew on that apple.
So I seem to have a delay in this presentation. Okay, so these are two things that, in my view, are people who are eminent ecologists who I look to as for mentorship from the previous generation of Dan and me. And when I looked on your website about this, I was delighted to find that both of these were distinguished visiting ecologists in past years. So it's nice to be in such august company. And I noticed after I did this that they're both named Jerry Franklin and Jerry Malilla. So maybe people named Jerry say wise things. But they actually said these things to me over a beer and a coffee, just, you know, the normal kind of bullshit at ESA meetings that we do. And one thing that Jerry Franklin said one time that really stuck home to me was that he thought nutrient budgets are ecosystems ecologists' most important contribution to ecology. Now that may sound pretty mundane, and you know, I mentioned this one time to a community ecologist, and they say that's why we don't like ecosystem ecologists. But there's something very, very important in that, and what Jerry was getting at was the idea that mass balance, which underlies why we do nutrient budgets and conservation of matter, sets very, very strong constraints on how a system can behave. So the input-output budget of a compartment, where the stuff comes from for the input and where it goes determines how those compartments are connected to other compartments or how ecosystems are connected to other ecosystems along the landscape. The fact that you cannot create matter or destroy matter in those input-output budgets puts very, very tight constraints on behavior of an ecosystem, and it means that only certain solutions are allowed, either in mathematical models or in nature. I remember a community ecologist asked me one time, and I forget who it is, and I'm not dumping on community ecology, I like community ecology, but a community ecologist said to me one time, I don't understand how you guys can be talking about nutrient budgets when we haven't yet figured out how communities work, and you're already going to the next step. And my response was, until you constrain the community by a nutrient budget, you won't be able to figure it out. And in fact, putting these constraints on what species can do because of the input-output budgets is actually going to make the problem a lot simpler in community ecology. So that's one word of wisdom that is going to flow through this talk. The other from Jerry Melillo is a little more poetic, even Zen master-like, and I think of Jerry Melillo as one of my Zen masters in my life, is nature is the orchestration of asynchronies. And Jerry never explained to me what that meant, but I kind of have chewed on that over the course of my career a lot and thought about it. And what the point Jerry was getting at is that things in nature don't happen all at once. One thing happens, then another thing happens, and there are delays in the transfer of matter and information and energy flow through a system, and those delays can result in complicated things happening. And what nature does is to orchestrate those delays and asynchronies and to get something that's more or less working and more or less stable, but we don't know how that happens. Okay? So I'm going to talk about that today in terms of wild rice, which grows throughout northern Minnesota, Wisconsin, and upper Michigan, and then into Manitoba. And here's a wild rice bed in a lake. It forms nearly monotypic stands. And here's some people harvesting wild rice in the traditional way. This is actually on a reservation that I've been working with on this problem. And on the edge of the stand here, maybe it starts to compete with pickerelweed and other things. But in the middle of the wild rice bed, it's nearly a monotypic stand. Here's a seed head of wild rice. It's actually quite a beautiful plant. This is right during the flowering stage. The male flowers up here and the female flowers down here. It's really quite lovely. And wild rice has great cultural significance in Minnesota. It's called Manoman by the Ojibwe, which kind of means Manoman. It means sacred food or communion host or something like that. But the Ojibwe have had real interest in this, both treaty rights and also in managing wild rice. And so this has been a nice opportunity to work with tribal members on a culturally important plant species, as well as just learning some science here. So these Indian bands, it's part of their traditional knowledge that wild rice beds go through roughly a 4-year cycle, boom and bust cycle. So they would know that if a lake is has a particularly good year, probably is not a good idea to come back to that lake the next year and let it rest for a couple years and then come back, and they'd go off. And basically during the wild rice harvesting season, they were nomadic and they would just visit different lakes, and they would know which lakes were in which parts of a population cycle. But various wild rice researchers have also described something that looks like population cycles. Here's some data from the 1854 Treaty Organization, which is a group of tribal biologists that monitor resources in northern Minnesota that are protected under treaty rights. Here's some inventory data from one of their wild rice lakes over the years. And you see there's roughly a 4-year cycle where a really productive year is followed by a crash and then a slow recovery. I'm not sure what's happening here. A lot of our lakes are kind of flattening out here. So this may be adjusting to climate change or whatever. So we're not quite sure what's going on, but even there you see kind of a cycle adjusting itself here. But there's something roughly like a 4-year cycle here. So we're interested in that. And some possible explanations for oscillations in plants really don't pertain to wild rice. One is age-dependent demographic process. Basically, an unbalanced age class distribution, and those of you who are foresters in this room know what that means. You just have a dearth of a certain age class that's going to move through the population, and it's going to cause cycles in the harvestable yield or the biomass of a plant. Aspen does this a lot. I think lodgepole pine does this a lot, so forth. But wild rice is an annual plant. So these age-dependent demographic processes don't apply. Competitive interactions, again, really don't apply. You can't get a limit cycle because of competition here because it's just largely monotypic stands. Climate, sunspots, and drought cycles might apply except for El Nino, which is roughly a 4-year cycle. None of these are on a 4-year cycle, and the fluctuations in wild rice populations, first place, don't seem to line up with El Nino and La Nina. Second place, they're not region-wide, like you would expect if this was caused by climate. That different adjacent lakes could be out of phase in their cycle, and even a large lake with a lot of bays, different bays can be out of phase with each other. So it's probably not something, a regional driver like climate. Herbivores, swans, ducks, geese, prey on the wild rice, but their populations never seem to be large enough to cause a predator-prey cycle between rice and their populations. And in fact, there doesn't seem to be any correlation, like a Lotka-Volterra predator-prey cycle between the duck population and wild rice. Parasites, there is a rice worm that at the very end, I'll talk about a few observations we've been making about that. But again, the rice worm doesn't seem to decimate. That doesn't seem to be a cause for the crash in the population. It doesn't happen after a big outbreak of rice worms. So that leaves the final idea, which is our hypothesis, is that litter accumulation inhibits the growth because of a delay in the release of nitrogen from the plant litter relative to the period when the wild rice needs to take up the nitrogen. So you might say, before I get into the wild rice biology of this, you might say, well, why should a delay in the supply of nitrogen cause a population cycle? A delay should just make things run slower, right? A delay just means lower nitrogen availability. Why doesn't it just decrease productivity? Why is it causing a cycle? And to show you why, just mathematically, you should all recognize this, the logistic equation here, except I put a delay right there in the logistic equation. So the growth of the population per time, the growth rate of the population right now is related not just to the size of the population now, but to the size of the population tau units before now. So tau units ago. And you could do this easily in Excel. So here's a homework problem for the students. Go home tonight and program this into Excel. And here's an Excel spreadsheet of the result to convince yourself. As I increase the delay, is increasing tau. When I have no delay, tau of 0, here's the nice S-shaped curve for the logistic equation. If I make the delay 1 year, it's an overshoot, but comes back down along the so-called carrying capacity. If I make it the delay 2 years, start to see some oscillations, but they're damped. But by 3 years, we have sustained oscillations. So mathematicians and engineers have long known that if there's a delay in the equation or there's a delay in the system or the solution of the equation could start oscillating like that. So the problem with these kind of models, which are called delay differential equations, is that the delay is put in by hand. There's no mechanism producing the delay. It's just put in there by hand. And that leads to some really interesting mathematics of delay differential equations, but doesn't give much biological insight into what is the mechanism behind the delay. So I wanted to really get at a biological mechanism behind what the delay is. And so this leads to our hypothesis, which I alluded to before, but here it is a little bit more strongly stated, that microbial uptake of nutrients from the sediments during the initial decomposition of the litter could delay delivery of nutrients to growing plants in the spring and thereby cause wild rice population oscillations. Now, the uptake of nutrients by the microbes while they're decomposing the fresh litter is because the carbon-nitrogen ratio of the litter is much wider than the microbial carbon-nitrogen ratio. And so for every mole of carbon the microbes take up from the litter, incorporate in their biomass, there is not sufficient nitrogen in the litter to go along with that. So the microbes actually have to import nitrogen from the surrounding environment. Many of you, particularly if you work with Dan, you know about this process. It's called nitrogen immobilization by the microbes. Where the delay comes in is that wild rice, being an annual plant, has to take up all the nitrogen it needs for growth that year, and particularly to make seeds for the next generation from the sediment and water that year. And not only that, wild rice takes up most of its nitrogen. Here's the uptake curve for nitrogen within a very brief window about 40 to 60 and really 50 to 60 days after seedling emergence. This is from an old paper done in Minnesota back in 1976. So this occurs, and seedling emergence occurs in middle of May, maybe at early May to mid-May. And by the middle of June or end of June, all the nitrogen, almost all the nitrogen needs, the plant has to take it up. And then it lives off that nitrogen the rest of the year and keeps translocating nitrogen to the growing tips and eventually puts it into the shoots. The problem is there was one paper done on nitrogen or a release of nitrogen from decaying wild rice straw, and that paper seemed to suggest that the nitrogen was not released until August of that year. So the nitrogen is being released way out here, but the plant uptake is occurring over here. So that asynchrony between uptake and release is causing a delay in the system. So if you imagine a really productive year, you got a lot of litter returned to the soil, it has a lot of nitrogen in it, plus the microbes are taking up even additional nitrogen during that first year of litter decay. They're outcompeting the plants for it. That litter is not going to start to release nitrogen until the end of the growing season or even the following growing season. So the idea is there's a crash, and then as that nitrogen that litter starts to enter the release phase, known in soil science as the mineralizing phase, in subsequent years, the population can then recover. So the first step in doing all this is, I like to do is just develop an exploratory model to see if the logic of my thinking is correct. Now this model is not meant to be super accurate or anything like that. It's just meant to see, look, is the logic of my thinking correct here, and am I on the right track? So it's a box and arrow diagram of nutrient cycling in a wild rice stand, and it's a set of difference equations, including rates of microbial uptake and release of nitrogen, plant uptake, return in litter, inputs, and leaching. So here are the boxes. Here's the plant nitrogen. Here's available nitrogen in soil and water. Here's the sediment. Here's the first year of litter and the second year of litter. There's inputs and outputs here from streams coming into the water here. The plants take up a certain proportion of the nitrogen. They die, return all of their biomasses the first year of litter. The microbes then take an additional amount of nitrogen up into that during the first year of litter decay. So here's the microbial uptake pathway or immobilization. And I use A for that as per capita microbial assimilation rate. The second year, this litter becomes second year cohort. All of the original nitrogen plus the immobilized nitrogen gets transferred down here, and now it starts to release nitrogen or mineralize it at a certain rate K1. The rest of it goes to sediment, and then sediment mineralizes at a second rate K2. Here are the equations for the model. It's basically just look at every box. Every arrow that's got an input, you add it in. Every arrow that's got an output, you subtract it off. Just difference equations. And here's the microbial uptake parameter here. So I put reasonable estimates for K1 and K2 and so forth from literature on this, and then I just varied A. And this is what happens. So here's where microbial assimilation is low, in this case 40% of the amount of nitrogen in the plant litter. And you can see there are some oscillations, but they become damped out. Where it's roughly around 0.6, we have sustained oscillations. If we go even higher, so really a high amount of nitrogen uptake by the microbes, 0.8, we get unstable oscillations and population crashes. Where I'm plotting here is plant nitrogen, because all the nitrogen ends up in the microbes. Basically what's happening is that microbial immobilization causes nitrogen to get stuck in that plant loop, and there's a delay to the release of back to the plants. If you don't like simulations and you're more mathematically inclined, we could just put all those equations in a matrix. And if you're not a mathematician, don't pay attention. If you're a mathematician, I hope this will satisfy you. This matrix has 5 eigenvalues, 3 are real numbers, 2 are complex conjugate eigenvalues, and these are the ones that give you the oscillations, complex numbers. And X and Y here are functions of the model parameters. So what we can do is, there's a theorem that says these will yield undamped oscillations when should be the square root of x squared plus y squared. There's a little PC to Mac conversion problem here is greater than 1. So we could just plug in the parameter values for everything else and solve for A to get this inequality, and it happens at 0.66, which is roughly what we saw in the simulation, giving us nice oscillations. So it's kind of a proof of the theorem that in a model like this, it should yield oscillations at a critical value of the parameter immobilization. So to go to the experiments, the first thing we had to answer, which wasn't exactly the first experiment we did, but conceptually the first thing we had to answer, was does nitrogen limit growth? And we did a whole series of experiments in the greenhouse and pot experiments and so forth on this. So I'm going to show you our latest, largest one. Phosphorus or light might also limit growth. So Lee Sims, one of my graduate students, who was listed as one of the co-authors on the first slide here, did a fertilizer shade experiment where he provided shade with shade cloths of 25% full sunlight and full sunlight, no nitrogen, 300 milligrams per plant, no phosphorus, 110 milligrams per plant, which from the literature is about the maximum uptake rate of nitrogen by the plants. It's a fully crossed factorial, and the nitrogen and phosphorus were added in parallel with that uptake curve that I showed you before from that earlier study by Grava and Raisinen. So it wasn't all just dumped in there once. We added it to simulate that growth curve of nitrogen uptake. So he did these in cattle tanks, stock tanks, in which he hung cattle grid wire, and from that were hung plastic bags that he put wild rice sediment in and grew individual wild rice plants. And then the bags are suspended in water in the stock tanks to keep them at a cool temperature. And here's a shade cloth over here. Here's Lee. And here's his experiment in which some of these plants are fertilized by nitrogen, some phosphorus, some both, some neither. And here's his growth data, total biomass at the end of the growing season. And you can see a Julian day at the final harvest. Here's the growth curve. I'll just talk about the final harvest here because all these other early ones are the same. The first two bars are shade control and full sunlight control. And you see shading and full sunlight had no effect on growth. So these plants are not light limited. The next two are phosphorus, shade in the phosphorus and full sunlight in the phosphorus over here, and phosphorus with shade had no effect. The next two are nitrogen, and nitrogen does have an effect on growth, increase of growth above shade and phosphorus by nitrogen. And in fact, in full sunlight, nitrogen increases growth even more, so there's a little bit of nitrogen-light interaction. And then if we add light, nitrogen, and phosphorus, that's where we get the greatest amount of growth here. But the primary limiting thing seems to be here nitrogen. And once you add enough nitrogen, then you can get growth responses to everything else. An interesting sort of side thing about this is he looked at relative growth rate, and these are just the data from nitrogen alone, phosphorus alone, and full sunlight and sunlight alone. And you can see nitrogen, the ambient nitrogen is in the dark, the added nitrogen is light, same thing with phosphorus and sunlight. Nitrogen increased relative growth rate, but phosphorus alone didn't, and light alone didn't. But phosphorus and light had no effect, but for different reasons. The nitrogen increased relative growth rate because the underlying components of relative growth rate, unit leaf rate, specific leaf area, and leaf weight ratio, nitrogen significantly increased two of them. Unit leaf rate and leaf weight ratio had no effect on specific leaf area. So if you increase two of the underlying components of relative growth rate, you increase relative growth rate. On the other hand, phosphorus had no effect on relative growth rate, but that's because it had no effect on any of the underlying components. But we knew the plants had taken up phosphorus because the phosphorus concentration in the plant tissues had increased. On the other hand, light had no effect on relative growth rate because it increased unit leaf rate but decreased specific leaf area and leaf weight ratio, and these decreases here compensated for this increase over here. So the interesting thing that came out of this experiment, which was not something we had originally planned for, was that there are two ways to uphold your null hypothesis. Actually, three ways. Number one, the plants don't respond to a nutrient because it doesn't take it up. Number two, doesn't respond to a nutrient because the underlying growth components don't respond, or it doesn't respond to a nutrient because the underlying components respond in compensatory ways. Whereas there's really only one way to respond to a nutrient, and that is to take it up and have at least two of your underlying components respond in the same way. So the response to nitrogen, while it was satisfying, was kind of boring in a way. The more interesting responses were the null responses in the end. And this is a paper that's coming out in Ecology. It was just accepted. So the second question is nitrogen in fact immobilized in wild rice litter, and which litter, the roots or the shoots? So we did a litter bag study of this over several years, put litter in litter bags. Here's root litter, here's shoot litter over here, and you can see there's a fair difference in what the appearance of the litter, and the root litter is just tougher. It's more fibrous than the shoot litter is. So decomposing these things over time, over 400 days here, well, about a year and a half, you can see the shoot litter here, this is shoot litter here, decomposes much faster than the root litter does over here. And these are fits of double exponential decay models to the data. Not only that, the shoot litter and root litter, they both immobilize nitrogen because of microbial uptake, but in different temporal sequences. What I'm plotting here is over time, incubation time in the field in these litter bags, over 800 days for the shoot litter and close to 500 days for the root litter. This is the nitrogen and phosphorus in the litter as a fraction of the original. So 1 is the original amount. Anything above 1 is more than nitrogen and phosphorus than the original, indicating microbial uptake of additional nitrogen. Anything below that is release. For the shoot litter, there was a brief period of very rapid uptake of nitrogen immobilization peaks, maybe a second one the following year, the second year of decomposition, but it very rapidly began to release it. Probably coincident with the timing of nitrogen uptake by the plant. The roots, though, immobilized nitrogen for a much longer period of time and even into the second year, and it was only well into the second year, at the end of the second year, that there started to be a net release of nitrogen from the litter. So it's really the root litter was our original thinking as a result of this experiment that is responsible for this delay, not the shoot litter. The shoot litter actually seemed to compensate in another way. Lauren Hildebrand then did her master's thesis where she looked at, well, what happens when we add nutrients to the environment? How does that affect litter decay? And she called that her site quality experiment, where she decayed a standard root litter or shoot litter from wild rice with different amounts of nitrogen, phosphorus, or control added to tanks. And a litter quality experiment where she grew plants with different amounts with nitrogen, phosphorus, or control and enriched their litter and then decomposed them without added environmental additions. And neither one of these things affected decay of root litter. Root litter decomposes slowly and nothing affects it. It's probably because of the fairly high lignin content of the root litter. The shoot litter, though, did respond to these things, and when phosphorus was added to the environment, decay became much faster, which leads us to suspect that maybe the microbes are actually limited by phosphorus. But the more interesting thing is that the nitrogen-enriched litter actually decomposed slower than the control or the phosphorus or the nitrogen and phosphorus enriched litter. And the reason why that happened, that seemed counterintuitive, because usually when we fertilize things, the litter is of higher quality. It's got a higher nitrogen concentration, and so it should decompose faster. And in fact, the CN ratio to litter went down. The problem was that the plants grew taller, like Lee showed, and in order to support that taller growth, the stems had to be more robust, and to be more robust, they had to have higher lignin content. So adding nitrogen and making plants grow better, which would happen in a high nitrogen year, actually produces not only more litter the following year, but it's poor quality litter. And Lauren was able to show that this litter actually immobilizes even more nitrogen than the control litter did. So now that we've verified the underlying mechanisms here in the model, now we can start to do some ecosystem experiments. And actually, we set these up first because these were intended to be long-term experiments. So to go back to our model to help it design our experiments, The obvious thing is to grow these plants, we could always have to grow the plants with sediment present, but grow them only with fresh litter, grow them only with litter that we've taken and decayed in a greenhouse to the point where it's now starting to release nitrogen, or grow it only in sediments and see if by manipulating the presence or absence of each of these pools here, we can alter the cycle, or at least the growth of the plants, of what's going on. So that's our first, to test the nitrogen delay hypothesis. We just grew rice with litter in different stages of decay. But there's an alternative hypothesis here too, and that is that the litter itself could just physically suppress seedling growth. So the seedlings have to emerge through the litter. So to test that, we planted in half of these tanks, and I'll show you the tanks in a second, were planted with seeds below the litter. The other half of the tanks were planted with seedlings grown so that they were already above the litter. So if there was a physical suppression of the litter on the seedlings, the plants that we gave a little bit of a head start to should outperform the plants that had to start from seeds and grow through the litter itself. So the experiment was done in 100-gallon stock tanks. There were 3 litter treatments. Again, no shoot litter at all, immobilizing fresh shoot litter, or mineralizing partly decayed shoot litter. 2 planting treatments, plant with seeds that have to emerge through the litter, or seedlings already emerged, and it's a factorial cross yielding 54 experimental units with 9 reps of each of the treatment combinations. And this is what it looks like. These are the tanks. Here's the wild rice growing in it. These spider webs on bamboo, that's just something we have to do to keep the plants from lodging over in the wind of a thunderstorm in the summer. They just, the spider webbing string just keeps them up, and they just bop up against the string and just stay upright. This is a drainage bucket. Right in the middle is you can see the beginning of a tube here that goes down through the tank, comes out, comes up into the drainage bucket. That tube keeps the water table in the tank constant, and we add water to bring it up to the top of that tube once a week from a nearby well. And we could get the input-output ratio of the tanks by sampling rainwater to get the input and sampling drainage water here. So we got the input-output budget of nitrogen, phosphorus, and water. The plants are growing in here. These net bags are on the seed heads. That was what we did the first year, but we also found that if a rice worm got into that seed head, it had a tendency to eat all the seeds in that seed head. So we've stopped doing that. Now we just collect seeds by hand as they ripen in these tanks. So this is the results of the first year, and this was part of the thesis of Rachel Walker, another one of the graduate students on that. And the different colors are whether the plant started as seed or seedlings, and this is their biomass at the end of the year. And there was no significant effect of whether they started as seed or seedlings on the final biomass. So the litter has no physical suppression effect on the growth or emergence of the seedlings themselves on the final biomass. On the other hand, the different kinds of litter did significantly affect final biomass. If we grew the plants with mineralizing litter, they grew the fastest because they had the highest nitrogen availability. Immobilizing litter grew about the same as with no litter, just sediment. Maybe a little bit better because it may be that the shoot litter started releasing nitrogen early enough that at least they got a little bit of nitrogen available to them. But in any case, the greater the release of nitrogen from the litter, the faster the growth rate, which was to be expected but satisfying. And here are some p-values for the effect of litter type on various things. What I just showed you is tank biomass, roots and shoots together, p of roughly 0.05, tank root weight 0.03, total plant uptake 0.002. We had resin bags in there to measure nitrogen availability, very highly significant effect of litter type on nitrogen availability, but they also affected seed biomass. So here is a little wrinkle on the problem. We were thinking about just these parameters here, total biomass and nitrogen cycling, but if the litter type and release of nitrogen from the litter also affects seed biomass, then nitrogen actually has 2 effects on the plant population. One through the biomass itself, but also another effect through seed production and how many seeds there'll be there next year to start the next generation. So to sort that out, in subsequent years, we wanted to sort out the feedback of N availability on population dynamics through seed production, as well as through delays in nitrogen availability by litter. So we removed all the litter, we re-randomized new treatments, and the new treatments were half the tanks are thinned to 30 plants, and half the tanks were allowed to germinate freely. So the thinning is not to simulate any management recommendation here. By thinning the plants to 30 seeds at the beginning, or 30 plants at the beginning of each growing season, that sort of mimics that we assume there's only 30 seeds produced and they all germinate. So what we're doing in the thinning is shutting off that feedback between nitrogen availability, which is affected by the litter, and seed production. The plants where they're allowed to germinate freely, that feedback between nitrogen and seed production is still operating, as well as the feedback between nitrogen and litter production. And half the tanks contain both root and shoot litter, and half the tanks we removed the shoot litter and left the root litter only in there. It would have been really nice if we could add a treatment where we only have shoot litter and remove the root litter. The problem with that is you remove the root litter and you stir up the sediment and you cause all kinds of disturbance. So unfortunately, we couldn't do that. So what we're looking at here is the effect of two different kinds, both of the litters, either both the litters together or root litter alone on production. And so here's the result. Some of these symbols are kind of light here, so I'll go through it. But starting in 2005 and go into 2010, and we're getting ready to have the 2011 data on here, here's their cycle. Starts out, goes up, all crashes, and then is starting to go up again. And it looks like from the raw data that it's recovering. In 2011 data is probably going to be up around here. And then these different symbols, these are standard error bars. It's root litter thinned, root litter unthinned, the dark circles or the triangles. And the light circles are root and shoot litter thinned and unthinned. So let me walk you through this. You can see the biggest effect of all the treatments is actually at the peak of the population, the peak of the cycle, not right after the crash or when the population is low. So let's just focus on this peak year here. So this is for biomass up here over time, and here's nitrogen availability over time, and you see that's cycling through exactly in parallel with the cycle of biomass. So both nitrogen availability and biomass are cycling concurrently here, which is something our hypothesis requires. So if you look up here, there's a triangle that's open. That's an unthinned root and shoot litter added, and a triangle that's closed is a thinned only root litter alone. So in this case, adding the shoot litter actually increases plant growth above the root litter here, because the shoot litter probably starting to mineralize nitrogen towards the end of the growing season. So it's adding a little bit of nitrogen back to the system there and boosts growth a little bit. There's a big effect between unthinned here and thin down here. So this black triangle here and this black circle here, this is unthinned up here, and this is thinned. And when we thin the tanks, in other words, essentially controlling seed production and shutting off that feedback between nitrogen and seeds, that had a big depression of biomass production. So this shows that these different treatments not only all cause a 4-year cycle followed by a recovery, but also they affect the amplitude of the cycle. And both the nitrogen seed feedback and the nitrogen immobilization biomass production feedback are operating in the system here. Here's just to show you the correlation between nitrogen availability measured with resin bags for the thin treatments, root litter treatments, unthin treatments, root stem and root litter treatments. This is the correlation between nitrogen availability and mean plant biomass. And you see there's a nice correlation here. So as nitrogen availability goes up, biomass goes up. R squares are all greater than 0.95. And you can even see the cycle here. So here's 2005, 2006, 2007, and the crash to 2008, followed by a recovery. And I haven't plotted the other several years on there yet. So the time sequence along this line here, the trajectory along this line here, actually is the cycle in the covariation of nitrogen availability and plant biomass through the cycle. So what about the seeds? What's going on there? Another part of Lee Sim's experiment was to look at reproduction, not just biomass growth of the wild rice, but reproduction. So he's looking at mean number of seeds per seed head on the plant, and here's the shade control and full sunlight control, and there's no effect on seed production by light. You add nitrogen, and you get an increase in production even if it's shaded or unshaded over here. So nitrogen actually increases seed production. Phosphorus has no effect relative to light here. And nitrogen and phosphorus alone, in this case, adding phosphorus did not increase seed production, well, maybe in full sunlight here compared with here. So phosphorus is secondarily limiting to seed production, but predominantly nitrogen. So after a high nitrogen year in which you're producing a lot of high biomass, you're also producing a lot of seeds. So let's think about what that means for the following year. The following year, that high biomass becomes litter the next year, and the proportion of seeds that survive to reproduction the following year actually goes down. So this is the production in the previous year that's become litter. When you get high litter, you got a lot of immobilizing litter up here. The seedlings, because of nitrogen stress, very low proportion of those survive to adulthood the following year. So what we're actually looking at here is reproductive fitness of the plants. So this is starting to get into some evolutionary questions here of what is happening during the course of the population cycle that the wild rice itself is inducing on itself. It's resulting in oscillation and reproductive fitness of the population itself. And again, you could see the cycle through here. Here's 2006 over here in the triangles, low litter biomass and fairly high seed production. 2007 is here, 2008, a lot of litter biomass, but the seeds next year hardly any survived. There was a population crash in 2008. 2009, very little litter was entering the soil, but much higher seed survival rate over there. So this leads to what we're thinking is now, which is more elaborated than when we first started this 7, 8 years ago. It looks like these population cycles are a result of both nutrient cycling processes and these demographic processes over here. This is essentially our original hypothesis. High nitrogen availability leads to high biomass. Root litter production goes up, and root litter, you can see, immobilizes a lot of nitrogen for a lot longer, decreased nitrogen availability. The shoot litter lignin goes up, which leads to greater immobilization, decreased nitrogen availability. But a year of high nitrogen availability, you get high seed production. What Lee also found is that actually stimulates rice worm growth on the seeds, and there's a little bit of a negative effect on seed production, but not enough to overcome the direct effect of nitrogen availability on high seed production. But here's the beginning of year 2. You got decreased nitrogen availability, but you have a very dense seed population, both because of the effects of nitrogen the previous year on seed production and litter production. So now you have decreased nitrogen availability, a lot of very high competition among these seedlings for that decreased nitrogen, and there's a very steep self-thinning curve going on here, and both of these things together lead to a population crash the following year. And so these population cycles in wild rice are actually now a result of an interaction between delays in the nitrogen cycle and the delays in the demography, the demographic cycle of the wild rice themselves, and both of them are mutually reinforcing each other. So our conclusions are that this is probably, maybe it's bragging a little bit, but I think this is the first clear example that plant chemical traits can delay release of nitrogen during litter decay, and then that leads to population oscillations. I haven't seen this suggested or reported anywhere else in the literature, and that tissue chemistry and growth allocations to shoots, roots, and seeds are what's linking population dynamics to nitrogen cycling in the wild rice. It'd be nice to see if this happens in other annual plants or even in other perennial plants. But perennial plants are buffered a little bit by this because they can store nitrogen in perennial tissues and then use it the following year. But nonetheless, I think even some perennial plants might undergo this kind of boom and bust cycle. So finally, I'd like to, few acknowledgments, NSF for funding this over an 8-year period. The Fond du Lac Band of Lake Superior Ojibwe for their collaboration, students from the Tribal College who helped us over the summer, and especially the 2 tribal biologists, Nancy Schultz and Larry Schwarzkopf. And then finally, Brad Dewey's and our wives, Mary Dragic and Susan Perala Dewey, who always came out and lent an extra hand during harvest time and counting seeds and fertilizing and everything else that we needed to have done. So there are unofficial co-conspirators on this project as well. So I'd be happy to answer any questions that you might have.
Does the rice worm ever spin out of control? I mean, called wild?
No, pardon? I'll repeat the question. Tom asked, does the rice worm population ever spin out of control? And the answer is, it doesn't seem to. There doesn't seem to be any reports of that by wild rice biologists and tribal biologists, mostly in the US or Canada. And wild rice is also grown commercially, and the commercial growers don't seem to worry about it too much. They don't spray for it or anything like that. I do have an undergraduate student now who's looking a little more closely at the infection rate of the, what is the rice worm actually looking for when it lays its eggs on the seeds? And he's looking at a bunch of things. Is it just simply plant height? Or is it seed weight, or is it seed starch? What is it? And it really does seem to boil down to seed nitrogen, because we've analyzed the seeds for starch. There's no correlation between infection rate and starch, plant height, number of seed heads per plant, size of the seeds, no correlation at all in that. At first he was depressed by saying, oh, I'm not finding correlations. I said, that's great. We're not finding correlations. That means we don't have to worry about that stuff. We can discard that hypothesis. And it really does seem to be boiling down to the amount of nitrogen that's there. Both the amount of nitrogen in sediment and the nitrogen content of the seeds, which are correlated with one another. So the rice worm adult is actually somehow detecting seeds with high nitrogen and laying the eggs there. And then the eggs, the larvae, just burrow into the seed. And you could see a seed that's been infected by rice worm. It's, not only is it empty, but there's a little round hole in it. It's just, you pick it right out. Yeah, Mike?
So this is probably the coolest nitrogen story I've ever heard. It's really interesting. What did the tribe think about all this? I mean, were they thinking that they might come out of this with something to manage, their...
Yeah, the tribe actually has been really great in all this. Their interest in it has been really ecological. And they want to harvest wild rice with a minimum amount of management. Partly for, I'll just have to say, spiritual reasons. When you talk to tribal members about wild rice, when I said before, it's like a communion host to them, I'm really not being sarcastic or facetious. It really is. So they think, managing the wild rice is somehow disrespectful. And they want to do it. They realize there's always some need for minimal management. Right now, they're trying to harvest pickleweed at a lot of the lakes that somehow it's, for some reason, taken over. But that's the extent of it. They don't want to add nutrients. They just want to know what's going on. And it's been really great to work with people who just want to know about the basic ecology of things. And the other thing is, the reason why they don't want to manage also is because they can actually sell their seed at a higher price. Because they could say it's hand-harvested wild rice from real wild rice lakes, not paddy wild rice, which is drained and the rice is harvested with a combine and it's fertilized and everything else. And they actually could, by not managing wild rice, can sell their rice in New York City for $12 a pound. Whereas commercially grown wild rice goes for $5 a pound in New York City. Now, they don't have as much wild rice to sell, but they could command a much higher price. So they've just been very happy about the knowledge that's gained from this. And the students, it's just been fun working with the students and actually getting the students to see that they might actually have a career in science. That, you know, they don't just have to get stuck on the reservation. But even on the reservation, the tribe just built a new natural resources management building with wonderful labs. I mean, they've got laboratories in there that are as good as my lab. And so they're really, the management part of the reservation is working with the tribal college to try and encourage students to go into science and learn some science and bring that science back to the reservation, either from medical school or biology or geology or hydrology or whatever, and help the tribe out more. And this project actually has been turned out to have been a showpiece for doing this. What's interesting is I actually had this idea when I first got to Minnesota. And so I called up the tribe and I said, hey, I'm from the university and I got an idea about wild rice and I'd like to work with you guys. And the person at the other end of the line says, we don't work with you bastards from the university, and hung up the line. And I thought, so I called back and I said, I'm sorry, maybe you didn't understand. They said, you're the guy from the university, right? And I said, yeah. And they said, what don't you understand about bastard? And hung up the line. So I met Larry Schwarzkopf, one of the tribal biologists. I just happened to see him at a local meeting. And I said, so what's this all about? And he said, well, actually, there's a lot of animosity between the tribes and the university in the cities because the agronomy department came up early on, 30 years ago, and said to the reservations, hey, let's work together on wild rice. But then what they did was they took the seed off the reservation and started the paddy wild rice industry. And the tribes got nothing. And the whole history of the treaties and everything else with the tribes is whenever white people say wild rice, we get kicked in the head. So he said, so what's your idea? So I told him, he says, well, that's a great idea. And I said, great, so we could start working on this. Let's write an NSF proposal. And he said, not so fast. He said, you got to get the trust of the tribe. And I said, all right, fine, I'll come down and give you guys a seminar. He said, no, that's white thinking. We don't work that way. I said, what do we do? He said, well, first place, we're going to want to see that you're going to stick around here for at least 10 years. That way we know that you're dedicated to the area. And we just want to see what you're doing. So if you got other things to work on, go do those. Eventually people will see and everything else. And he says if trust builds up, it'll happen. And if it doesn't happen, it doesn't happen. I said, well, okay. He said, look, I'm not against it. I'm all for it, and I think it'll happen. It's just going to take a long time. So, okay, Larry, I'll trust you. And so then I went and did other things, moose things and everything else. And then somehow I got involved in the preservation of an old-growth forest in Duluth that there were rumors that it had sacred status to the tribes. And I'd go before city council meetings and planning meetings and all this stuff and try and explain the biology. And there was always tribal members in the back of the room watching these whiteys discuss their sacred forest. And they never said anything, and I figured, well, I don't know whether to say anything to them or not, but I'll just let them talk to me if they want to, and if they don't, okay, that's fine too. And they never said anything to me. But then one day Larry calls me up and he says, so I'm hearing a lot of good things about you defending the forest. And I said, well, Larry, you know, it's just kind of my job and my passion. And he says, well, remember I said before that it's going to take about 10 years to build up trust? And I said, yeah. He says, well, it's been about 10 years. He says, I think people trust you now. And that's how it happened. And so that's how I got basically permission with the tribes to collaborate with them. So it taught me a big lesson. When you're working with a different culture, there are huge cultural hurdles to get over. Huge. That we're not even aware of in our Western culture here. And you have to get over those cultural hurdles first. And it's not like they're wrong and you're right, but going to them and saying, I got NSF money and let's do research together, which is like mom and apple pie to us. To them, it's like, we don't care. We don't care. That's not the big thing. So that's kind of a long story, but it's an interesting story. And if anybody's thinking of working with different cultures, but particularly Indian tribes, I think that it's a learning lesson. And what I've also learned is just because you got the trust of one reservation, that doesn't mean you got the trust of the next reservation down the road. You got to go through it all over again with them as well.
One more
So
question for John?
it's learning to work with the tribes in a different culture on this is in some ways been the biggest learning experience of the whole thing. Yeah.
You're working in wetlands, right?
Yeah.
You never mentioned anything about water table variation or low redox potentials and denitrification or anything.
Okay.
Like that. I'm just curious. how
Well,
that figures into your story.
yeah, water table fluctuation is often, it does affect the rice, but only in the early growth stages when it's coming up. After that, once the shoots are emergent, water table fluctuations don't affect it. But the water table fluctuations don't seem to bear any relationship to the 4-year cycle. In other words, a productive year is not because of any particular water table position. So there doesn't seem to be a correlation there. Although high water table can drown the seedlings and cause a crash that year. For denitrification, there's just no nitrate in these systems to be denitrified. There's no nitrate coming off the upland. We've rarely seen nitrification in any upland soils throughout northern Minnesota. And there's no farms around there for nitrate runoff from the farm. So we haven't measured denitrification, but we've never seen any nitrate to denitrify at all. So I suspect that it's probably zero or close to it.
We've got John here for another day. There's a little bit of time open tomorrow where you can join in.